Hello everyone. Today I'm excited to take you on a journey into the fascinating world of genetic engineering, specifically focusing on a process known as bacterial transformation. My name is Ruoxuan Wang, and in our Biology 110 course, we've been exploring the incredible ways scientists manipulate bacterial DNA to unlock new possibilities in medicine, agriculture, and beyond. Before we dive into the specifics of this experiment, let's establish some key background information. Genes, the DNA segments that serve as the essential functional units in hereditary transformation, play a crucial role in the inheritance of traits from one generation to the next. Our protagonist is E. coli, a prokaryote, a unicellular organism without nucleoids or membrane-bound organelles. Our journey takes us back to the groundbreaking work of Griffiths, who observed the transformation of non-pathogenic bacteria into pathogenic bacteria. His discovery showed the mysterious influence of an unknown heritable substance from the dead S cells. This substance empowered R cells to produce capsules. Cell transmission is the crux of our experiment, where cells pick up foreign DNA from their surrounding environment. To achieve this, we work with competent cells that possess the remarkable ability to pick up foreign DNA. Now, let's introduce the key player, the plasmid, a small cellular DNA found in bacteria, carrying unique genes that code for distinct proteins, giving cells unique characteristics. Critical features within the plasmid include the origin of replication, where DNA replication initiates, and the multiple cloning site, hosting various restriction enzyme sites for genetic manipulation. The selectable marker, a gene producing a protein, empowers transferred bacteria to survive in conditions where untransformed cannot. Now, let's explore the practical aspects of this process and witness the magic of genetic engineering in action. Let's see my hand drawing for illustrate the principle. Scientists often use antibiotic resistance genes as selectable markers. We know only cells that have acquired a plasmid will be able to grow. The plasmid used in this step contains the gene for ampicillin resistance, also known as AMPRG. However, when the cell contains a plasmid carrying the AMPRG, it can make the enzyme beta-lactamase and break down the ampicillin before it affects the cells. We will start with E. coli, which are sustainable to ampicillin because they do not have a plasmid with AMPRG. The bacteria have been growing on algae agar. In this lab, we will transfer some of the bacteria with a plasmid called PUC19. PUC19 plasmid contains AMPRG and LACZ, which encodes the beta-galactosidase enzyme. Instead of galactose, we will use IPTG in this experiment. And also for visual screening purposes, we use X-gel, which can make the clonized on the plant blue. Let's see the procedure of this lab experiment. We first label 2 tubes, plus and minus and agar plates according to plasmid content. And then we saw chemically competent cells on ice. Transfer cells to label tubes and add plasmid to the plus tube. Then we incubate tubes on ice, move to a 42 Celsius degrees water bath and return to ask them. We add LB and stick plates tape and incubate upside down. Now, the moment we've all been waiting for, the results of our bacterial transformation experiment. Behold a snapshot of our plates. In the LBAMP plus PUC19 plate, we observe blue colonies. Moving on to LBAMP minus PUC19, there's a lack of colonies. Now, look closely at LB plus PUC19 and LB minus PUC19 plates. Both showcase light yellow colonies. These results are in line with our expected ones. As we transition from the laboratory setting to the real world, let's explore the profound impact of bacterial transformation on biotechnology, medicine, and agriculture. Bacterial transformation, as we've seen in the lab, is a game changer in biotech and medicine. Introducing specific genes into bacteria allows us to produce recombinant proteins, vaccines, and medicines. Insulin, vaccines, and growth hormones are just a few examples. Now let's address a global urgency: antibiotic resistance. Superbacteria armed with resistance genes pose a severe threat. Our experiment delves into understanding how bacteria acquire these genes. This knowledge is a critical step toward developing new antibiotics, combating the risk of superbacteria, and safeguarding global health. In the vast fields of agriculture, genetic engineering reshapes the way we grow our food. With bacterial transformation, we create crops with enhanced traits such as pest resistance and improved nutrition. Look at efficiency. Genetic modification significantly reduces the waiting time compared to conventional breeding. This method is key in crafting genetically modified organisms, also known as GMOs, that address the challenges of modern agriculture. Before we wrap up, I want to express my sincere gratitude to those who have been important in this journey. Firstly, a heartfelt thank you to Professor Li for your guidance and support. Your expertise and passion for biology have been truly inspiring. To my parents, your support has been the bedrock of my journey in learning biology. You have fueled my pursuit of becoming a molecular biology scientist, and for that I am truly grateful. I also want to express my thanks to my incredible SigPair group in our BIOL110 course. Our discussion really provides me with valuable insights. Finally, to everyone watching, thank you for your time and interest. Your processes is the final piece that make this biology journey complete.